hope everyone is doing well. At this unit, we will be talking about carbohydrates. I am Karima, assigned for the materials, procedures, and data. My co-members are also with me. Dana is assigned for the objectives, Ara and Micah for the pre-laboratory, Mark Raywin and Mira for the post-laboratory, and for the science behind in this video will be explained by Mark Raywin, and our video editor will be Micah. Humans can eat anything in order to satisfy its needs and able to survive. Same true with animals. But how about plants? Have you ever wondered how this living matter able to survive? Primarily, it is because of photosynthesis. Photosynthesis is a word that came from two Greek words. Photo means light and synthesis means putting together. Thus, photosynthesis means using light to put things together. It is the only process wherein plants are able to make its own food, which is glucose, with only the help of carbon dioxide, water, and sunlight. Not all glucose that the plant produces are all used up. Some extra glucose are stored by the plant for later use. At night, starch, which is produced by photosynthesis in daytime, is converted to small soluble sugar and is transported to the root through sieve tubes. The food that they make is the polymerization of glucose molecules, what we call starch, otherwise called as amylin. It is the most common carbohydrate in human diets for people characterize themselves as vegetarians. This is another episode of Biochemistry at Home, titled I'm Hurting Baby, I'm Broken Down. Keep watching until the end. To prove that plants contain starch, we will be using the starch test. It is a chemical reaction that is used to test the presence of starch for iodine. The appearance of blue-black pigments is the combination of starch and iodine. The process is using the insulation or the aluminum foil method to be pasted on the leaves at dawn as recommended. The materials needed are the following: normal plant, variegated plant, ethyl alcohol, povidone iodine, aluminum foil, scissors, fork, pan and gas stove, hardbound glass, and a transparent sheet. In a plant with normal green colored leaves, paste light insulation in one of its leaves for at least 48 hours before the laboratory experiment. In another plant with variegated leaves, paste light insulation also in one of its leaves, same time frame with the fork. Using the scissors, gather the leaves from the normal and variegated plants, both insulated and non-insulated. Remove the insulation gently and mark the insulated leaves as we identify it later. Boil 2 glasses of water in a pan, then put the insulated and non-insulated normal leaves. Let it sit for 60 seconds and remove the leaves from the pan. Glass, fill it with ethyl alcohol and put the alcohol, remove and dry a little bit the chlorophyll leaves and place it in a transparent sheet. Replace the used ethanol and repeat the process using the insulated normal leaf. Using the povidone iodine, pour and spread a portion into the two dechlorophylled leaves. Focus on its color changing. The non-insulated normal leaf must appear blue to black, reflecting that it contains starch, while the insulated normal leaf must be brownish in color, insisting that it contains no starch. Document the data. Clean the materials and repeat the whole procedure, but this time using the non-insulated and insulated variegated leaves. Document the data.
In our experiment, we observed the color changes on the chlorophyll leaves until the iodine was added. We used two types of plant leaves, the normal one and the variegated or the white pigmented plant leaves. In the normal non-insulated leaves, small bluish-black pigments are observed, an indication that there is presence of starch, while in the insulated leaves or the leaves covered with aluminum foil showed brownish color pigments indicating that it contains no starch. On the other specimen, the variegated leaves, it appeared the same observation with the normal leaves, but showed little effects due to lack of presence of chlorophyll. Non-insulated variegated leaves revealed little bluish-black pigments, while the insulated variegated leaves revealed a brownish color. Starch is a homopolysaccharide that contains amylose, which is in linear form, and amylopectin in branch form. Amylose molecules consist of a single, mostly unbranched chains of glucose molecules, shaped like a helix. It is speculated that iodine gets stuck in the coils of the amylose molecules. The starch forces the iodine into a linear arrangement in the middle group of the amylose coil. There is transfer of charge between the starch and the iodine. This changes the electron arrangement and hence the spacing between the energy levels. Thus, the new spacing absorbs visible light selectively and give the complex its intense blue color. This changes of color is caused by change transfer or CT complexes.